Hello everyone, and welcome to today's webinar, Biopharma 101 Analysis of mRNA-based Therapeutics. I am 𝗕𝗫𝗍𝗫 of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by SCIEX. To learn more, visit sciex.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speakers, Stephen Calciano, Market Development Manager, Biopharma MS at SCIEX, Paula Orens, Market Development Manager, Biopharma CE at SCIEX, Nathan Condor, Workflow Specialist, Nucleic Acid Therapies at Cytiva, and Dave Sokolowski, Global Workflow Manager at Cytiva. Stephen, Paula, Nathan, and Dave, you may now begin your presentation.
Hello everyone, and welcome to Biopharma 101, Analysis of mRNA-based Therapeutics. My name is Stephen, and I'm a Market Development Manager for Biopharmaceuticals at SCIEX. I'm joined by my colleagues Paula and Nathan, and we're here to discuss mRNA workflows with you today. So first, let's start with an introduction on this topic of mRNA delivered by Nathan, and then Paula and I will take a deep dive into recent advances in analytical capabilities to support these next-generation modalities.
Hello, this is Nathan Condor, Nucleic Acid Therapeutics Workflow Specialist for Cytiva, and today I'd like to give you an overview around mRNA workflows. So here's an overview in general of the mRNA production workflow for manufacturing, starting with plasmid production, going into the second section, mRNA manufacturing, and then going into encapsulation and filling with LNPs as the final drug product. But for this section, we're going to focus on plasmids. Plasmid production is utilized for several different biomolecular applications, including the production of mRNA. So for those that don't know, plasmids were discovered in the mid-1900s. They've been around for a while in biotherapeutic applications and other biological research applications. They're small pieces of double-stranded circular DNA that exist in many prokaryotic and a few eukaryotic cells. They replicate independently from the bacterial DNA and are inherited by the progeny bacterium. Through genetic engineering, you're able to insert a gene or sequence of interest into a plasmid and then have the bacteria start to produce this modified plasmid, typically called a vector, biologically in cell reactors. That material, the plasmid vector material, is then separated from the cells, bacterial cells, and purified to produce up to GMP grade material for therapeutic applications. In the case of mRNA, the plasmid will be linearized then to act as a template for the mRNA production enzymatically through in vitro transcription or IVT. But plasmids may also be used to secrete proteins from the cells. The proteins will then be purified later as a therapeutic agent, as in mAbs, or they may be used as a direct DNA vaccine in some cases, or viral vector for gene therapy. Some of the appeal around mRNA is that there's a lower risk of cell-related impurities. And the titers have been higher than some traditional biomolecules like mAbs. This means that you can get a lot more active material out of a certain comparable volume, and you don't have to have these huge 2,000-liter bioreactors to produce enough material for a disease population size or a vaccine population size in some cases. Now let's take a look at a few scenarios for scale-up to manufacturing. Due to the urgency of the pandemic, many of these processes were scaled up quickly using equipment that was useful for production. However, oftentimes the equipment wasn't designed with with mRNA in mind. And with the higher titers of material produced, you will be able to use relatively small volume reactors. You can deal with they were dealing with low efficiency, still capturing enough material to deliver a large number of doses. So in the design, you definitely don't want to end up with a factory with these huge volume capability when you're looking at rare disease or more of a small personalized space, because you can produce a relatively high amount of material with a small volume. So currently there is no definitive platform for mRNA development and production. The unit ops can vary widely. There can be some homebrew methods that don't scale well, but in general, you can group them into these following subgroups. Cytiva and Paul have a good presence with bioreactors, filtration and purification products in these DNA template and RNA product production, and in LNP further polishing. And then Precision Nano contributes a considerable amount of expertise around the LNPs themselves and lipids in general. And then Vanarex fills a need here for filling, and other opcos like SCIEX for analytics. Analytics is something to be considered right away, as many steps don't have in-process monitoring or analytics currently with some of these homebrew methods. So material can sometimes be over-consumed, allocating to many different steps in purification and then analysis and filtration and analysis and so on.
Thank you, Nathan, for the great introduction and for the state of the industry of mRNA production. As you mentioned, analytics is something to be considered early on in the mRNA development process. So today, we'll be providing an overview of 5 core workflows that can help you better characterize and monitor your mRNA-based products. We will cover plasmid analysis, RNA sequencing and shape modeling, mRNA purity and integrity analysis, identification of the 5' end structure, and lipid nanoparticle impurity assessments. Plasmids, which are double-stranded DNA, are used throughout the development of next-generation biopharmaceuticals. Analysis of double-stranded DNA is an integral part of manufacturing nucleic acids as therapeutics for gene and cell therapy, as well as for DNA vaccines. It's important to ensure their quality, as plasmid production can generate topological variants, including supercoiled, open circular, and linear isoforms of varying length. The Food and Drug Administration guidelines recommend manufacturers to include the gene map with relevant restriction sites for gene therapy vector constructs and vector diagrams identifying the gene insert, regulatory regions, and pertinent restriction endonuclease sites when submitting applications for investigational new drugs. This requires an accurate sizing workflow, and capillary gel electrophoresis with laser-induced fluorescence detection provides an easy workflow for both topo-isoform analysis and accurate sizing with high sensitivity. Plasmid isoform testing can help ensure product quality throughout all stages of development. Monitoring plasmid isoform distribution during production can help you optimize each of the bioprocess unit operations efficiently. Traditionally, this assay was run on agarose gel electrophoresis, and the image on the right shows an example analytical result. Lane 1 on the left shows a molecular weight standard, and the lane on the right represents the supercoiled or covalently closed circular DNA, linear DNA, open circular DNA, and a multimer from bottom to top. The quantitative performance and sensitivity of this assay, however, can be improved through capillary electrophoresis technology. Capillary gel electrophoresis is a high-sensitivity plasmid DNA sizing workflow. Here we show the conformation of different isoforms with control standards. Starting with the lowest trace in purple, we see a 5 kilobase plasmid preparation sample. Above it in blue is a linear plasmid DNA control standard. In pink is an open circular or nicked control standard, and in green is a combination of the plasmid prep with both control standards. We can see that the plasmid prep contains a high proportion of supercoiled DNA. but there are some impurities, including a significant amount of open circular DNA. Here we can demonstrate the workflow sensitivity by analyzing a double-stranded DNA test mix. The graph on top shows the analysis of this test mix with a starting concentration of 404 nanograms per milliliter, down to a limit of detection at 0.79 nanograms per milliliter by capillary gel electrophoresis. The bottom graph shows a sample containing the sample DNA test mix, but also includes related product fragments. This method provides high resolution between these complex plasmid DNA samples. In general, CE methods are highly repeatable, and this capillary gel electrophoresis workflow for plasmid analysis is a good example of that. Data from 8 injections of the 1 kilobase test ladder at 20 nanograms per milliliter is shown on the graph. The percent RSD of the 1,636 base pair fragment was 0.43% for migration time and 2.19% for corrected peak area across the 8 samples. Analysis of restriction fragments can help further characterize plasmids to map genes, inserts, and regulatory regions. This graph shows analysis of a sample containing restriction fragments of a bacteriophage DNA, a DNA standard ladder, and a linear plasmid DNA. This method offers high resolution between closely sliced DNA and can be used for accurate size determination of unknown fragments. Now moving on to our second analytical workflow, RNA secondary structure modeling studies offer an analytical solution to accelerate the commercialization of RNA-based products. Capillary electrophoresis technology enables an automated, high-throughput approach to map RNA molecules and understand their secondary structure and provide product-specific confirmation through base identification. The workflow we'll discuss in this section is RNA SHAPE analysis by capillary electrophoresis. Let's take a look at some of the complexity we can be dealing with when analyzing RNA. Here is a view of an example RNA, 𝗕𝗏1 retrotransposon. You can see that just having sequence information of this molecule is only telling part of the story. There are complex structural elements throughout the RNA, including internal loops, hairpins, multi-branch loops, bulges, and in this case, even dimerization as seen through the symmetry in the middle of the molecule. RNA chemical probing using the SHAPE reaction depends on RNA 2' O-adduct formation following RNA acylation by highly electrophilic reagents, such as NMIA. Unconstrained mRNA contains sites that are readily accessible for this reaction. The degree of RNA acylation over the RNA transcript is critical for premature reverse transcription and cDNA enrichment. Using cDNA fragment analysis by capillary electrophoresis and computational software, the RNA SHAPE reaction can be used to perform RNA secondary structural modeling. The overall RNA SHAPE workflow is depicted in this diagram. Starting with the RNA we want to analyze, we split the sample into a control reaction and a SHAPE reaction sample. The control reaction provides us with the baseline RNA structure to compare to the NMIA reacted samples. Reverse transcription using IDT freedom dyes is then carried out in the presence of a dNTP mix. The resulting cDNA fragments and sequence ladders are then analyzed on the SCIEX 𝗕𝗓𝗄 genetic analyzer. We then perform data interpretation to assign nucleotide reactivity values that are found by comparing the SHAPE reacted samples with the control samples. These reactivity values are then used with structural modeling software to enhance structural modeling accuracy. The example diagram on the right shows how structural modeling with SHAPE can offer insights into RNA secondary structure that could be missed when doing in silico modeling alone. Here's a summary of the sample preparation that's required for SHAPE analysis. On day 1, we relax the RNA structure and then perform the RNA acylation reaction with the NMIA reagent to yield the RNA 2' O-adduct formations at flexible RNA regions. Then we'd perform RNA precipitation. On day 2, we synthesize the complementary DNA fragments with reverse transcription of both the positive reaction and our negative reaction. Step 2 combines both the positive and negative reaction samples and creates the mixture. On day 3, we add in the DNA ladder and combine all of these together and run shape analysis using the 𝗕𝗕𝗃𝗵𝗿 fragment analysis system. The nucleic acid fragments are resolved one base at a time, and the fluorescence intensity at each of the complementary DNA fragments will indicate which bases are at the truncated region. Here's an example of 𝗕𝗵𝗿𝗃 RNA SHAPE reaction profiling by capillary gel electrophoresis. 4 primer sets were designed to cover the 1,395 nucleotide 𝗕𝗵RT1 transcript. Positive reactions indicates 𝗕𝗵RT1 transcript is treated with the NMIA reagent. Here, the blue signal indicates a positive reaction, and a green signal is a control reaction. The black and red traces are used as DNA sequencing ladders. Once capillary electrophoresis analysis is completed, ShapeFinder software is then used to process the data. The top section shows the normalized positive reaction in blue over the control reaction in green. The gap or difference between the blue and green trace suggests the degree of RNA 2' O-adduct formation, which can be interpreted as the flexibility of a respective nucleotide. The bottom section shows DNA sequencing for thymine in black or adenine in red. The lines in the diagram indicate the identity of each peak and their alignment to the 𝗕𝗵RT1 reference sequence. The figure on the left is a secondary structural model using the data processing tool RNA-Thor. You can see an example of the 𝗕𝗕𝗃𝗵𝗿 data that was analyzed with ShapeFinder that was then uploaded to RNA-Thor. RNA-Thor results include nucleotide sequence identity and a calculated shape reactivity value. These shape reactivity values can be used to optimize in silico structural modeling of RNA with real-world data points. Here is how all of the various workflow components fit together. First, you generate shape fragments and separate them by capillary electrophoresis. Then you generate nucleotide reactivity values with ShapeFinder that can then be used to feed structural modeling tools and improve in silico predictive modeling with real-world data. Our third and final CE-based workflow is used to determine mRNA purity and perform a size estimation. When optimizing the production of mRNA, it's important to assess the quality of the final product, including its integrity and purity, to ensure product efficacy. An effective mRNA sizing workflow can help resolve closely related fragments that differ by as little as a few bases, from complex formulations including mRNA encapsulated in lipid nanoparticles, CRISPR gene editing systems, and viral vector genomes. The key workflow we will discuss in this section is capillary gel electrophoresis with laser-induced fluorescence. Here we are showing the workflow for analysis of messenger RNA lipid nanoparticles. From left to right, we can see sample preparation of messenger RNA lipid nanoparticles were treated to release the mRNA for capillary electrophoresis analysis. In this example, we took a collection of samples from different storage conditions for a product stability assessment. The control sample preparation was diluted in water and heated at 70 degrees C for 5 minutes. The experimental buffer was a 0.3% Triton X100 buffer in water and heat for 70 degrees C for 5 minutes to release mRNA from lipid nanoparticles. Our studies showed that this experimental buffer did a much better job at extracting RNA from lipid nanoparticles than by simply using water and heat. Once the mRNA was treated with the Triton X100, the samples were loaded into a CE system and analyzed with a high-throughput kit for analysis. On the right, we can see an example of the analytical results we can expect from this workflow, which includes a main peak, a minor peak to the left, and a very low level of impurities. In the next few slides, we will show how we can analyze various mRNA lipid nanoparticle product storage conditions with this method to determine optimal conditions which can preserve mRNA integrity. Here we can see a profile of in vitro transcribed RNA prior to mRNA-LNP formulation. This data will serve as the baseline profile that we can use for stability and purity profiling over different product storage conditions. The green trace represents a blank sample, or our background signal. The red trace represents the IVT reacted mRNA, or in vitro transcribed reacted mRNA. The main product migrated at 17 minutes, corresponding to an expected size of 1.8 kilobases. We will show how the capillary electrophoresis platform can perform this accurate size estimation in a few slides. We also see RNA degradation products, either by natural degradation or incomplete in vitro transcription, labeled as impurities under the black bracket. We can also see a minor product right before the main peak. Also, notably, there are no high molecular weight impurities observed in this control sample. Here we can see an overview of the capabilities of this method for characterization of the starting material. Here are 3 electropherograms showing RNA profiling of 3 sequential injections. We can see the summary data in the table on the right across these 3 injections. The main product corrected peak area percentage was 89.28 with a %CV of under 2%. The minor product corrected peak area percentage was 5.61% with a %CV of less than 3%. The column to the right shows the calculated size of the main product, with an average size of 1.849 kilobases with a %CV of less than 2%. To determine the size of the RNA, we can utilize an RNA ladder. Once the RNA ladder is resolved, in this case over the range of 50 to 9,000 bases, the capillary electrophoresis software can then be used to determine size of unknown fragments within this range. This slide shows the analysis of both in vitro transcribed RNA and RNA extracted from lipid nanoparticles by capillary electrophoresis with and without oxidation. This graph overlays 5 electropherograms. The red trace shows the profile of free mRNA after in vitro transcription, which is also our baseline indicator for the expected level of impurities and high molecular weight products. The green trace represents mRNA lipid nanoparticles after synthesis. Pink and blue traces show IVT reacted mRNA and RNA extraction from LNPs subject to oxidative stress. The aqua traces are control. We can see in the green trace that the extracted RNA from the lipid nanoparticles resulted in a main product that has the same migration time as the baseline main product in our red trace. This tells us that the main product is still found in the mRNA lipid nanoparticles. We do, however, see in the green trace that we can detect a high molecular weight product. You can also see the free mRNA in the pink trace, which was exposed to oxidative conditions, is very similar to the aqua trace, which shows that the oxidative conditions degraded a majority of the mRNA. We can also see the blue trace shows a major decrease in the main product, which indicates that the lipid nanoparticle provides some protective effect for mRNA. This analysis can prove useful for developers looking to understand how their mRNA may be impacted by adverse storage conditions. This workflow can be used to determine the concentration of mRNA extracted from lipid nanoparticles. The x-axis on this chart illustrates the mRNA concentration, and the y-axis indicates the corrected peak area as determined by capillary electrophoresis. Using capillary gel electrophoresis with laser-induced fluorescence was analyzed with triplicate injection. We are able to determine RNA concentration over 3 orders of magnitude with a lower limit of detection of 97.9 nanograms per ml and a limit of quantitation at 390.6 nanograms per ml. Here is an overview of the workflow for automatic RNA size estimation. Panels A, B, and C show 3 sequential injections of RNA extracted from lipid nanoparticles for a sample that was stored for 92 days at -20 degrees Celsius. We can see the profile of the RNA ladder, which is in red, and the mRNA extracted from the lipid nanoparticle, which is in green, was consistent across 3 injections. The main product is labeled 1, the minor product labeled 2, and the higher molecular weight product labeled 3. Using data processing features in the capillary electrophoresis software, we can see the main product has an average size of 1.937 kilobases with a %CV below 2%. The minor product has an average size of 1.841 kilobases with a %CV less than 2% as well, and a high molecular weight product average size of 2.407 kilobases with a %CV of less than 4%. Capillary electrophoresis is a great technology that can be applied to analyze mRNA in many different ways to support product development and quality control. Now I'd like to hand it off to Paula to take us through some mass spec-based characterization workflows for mRNA and LNPs.
Thanks for the introduction and the handoff, Stephen. So I am going to cover our final 2 workflows, beginning with the identification of the 5' cap structure of in vitro transcribed mRNA. When mRNA is transcribed in vitro, or IVT, the 5' cap is necessary to maintain the biological function of the mRNA. This is typically done enzymatically in 3 reactions and results in a single nucleotide addition at the 5' end, depicted here in this nice animation. However, this 3-step process also results in an uncapped mRNA as well as various intermediates. And these intermediates can be observed throughout the process. The process, which starts as an uncapped mRNA with 2 phosphate groups at the 5' end, then the first enzymatic reaction forms what is called a G-cap, G based on the enzyme. The second reaction forms cap 0, and then the final reaction finally forms the 5' cap of interest. And I'll kind of just give you a second to observe these reactions and mechanisms without trying to explain it myself. As a control in this experiment, an uncapped sample was prepared and separated in parallel with the capped sample. So here on the left side is the full scan TOF-MS data for uncapped in blue and capped mRNA in black. The charge states of the main species in each sample are indicated by the red bars, or also hypermasses. And then on the right-hand side is a zoom in of the most abundant charge state, which is Z-11. And it's depicted as raw data as well as the reconstructed data, which exhibits great comparability between the 2. So you can see the diphosphate denoted here with 2 P's, this peak right here, containing version, or this version contains the most abundant as the uncapped sample, whereas for the capped mRNA, cap 1 is actually the most abundant peak, and that is exactly as you would hope it would be. We can also look at the XIC, or extracted ion chromatograms, of the most abundant charge state for the PP uncap, which is that double phosphate, and the cap 1 of the uncapped and capped mRNA respectively. Under the green umbrella, the reconstruction of the raw data allowed identification of different capped and uncapped species, as well as their metal adducts. Now, these adducts, sodium, potassium, and iron, were observed for mRNA carrying 2 or 3 terminal phosphates due to the strong chelation effect of diphosphate and the metal ions in solution without EDTA. Now, all species are showing excellent resolution for raw and reconstructed data, and there's great comparability again between the reconstructed and the raw data. This table lists all the species we were able to identify. As expected, we identified the mRNA intermediates from the 3-step enzymatic process of adding the 5' cap, as well as different levels of phosphorylation and those previously mentioned metal adducts. We are also identified in both capped and uncapped species. Now, to assess capping efficiency, the intact quantification workflow in SCIEX OS was used. And in brief, raw TOF-MS spectra was reconstructed and mass peaks were integrated using the analytics module. Now, a customized formula was used to calculate peak area ratios of each species against the total sum. And then all adducts were taken into account for the highest accuracy. And through this, we were able to determine that the capping efficiency was 43.2%. So a little less than 50, but not so bad, I guess. With this last topic, we're nearing the home stretch, and I might be a bit biased, but I think we definitely saved the best for last. No offense. In this workflow, we're covering oxidative impurities from ionizable lipids used in lipid nanoparticle formulations. Now, this is an extremely important topic because it was recently published that certain oxidations of ionizable lipids lead to covalent modifications of ribonucleotides, which results in a loss of mRNA potency. MC3 for short is an ionizable lipid used in LNP formulations. Like other ionizable lipids, its structure contains a tertiary amine, which is on a 3-carbon head group that is bonded via an ester linkage to 2 separate but identical alkyl chains containing double bonds at both C6 and C9. Now, on the bottom is a preview of the information you can gather when using electron-activated dissociation. This is the main peak of the ionizable lipid. And if I can call your attention to the percent intensity on the left-hand side, the main peak we see is 100%, but it's when you start to zoom in on the baseline to see what else is present that these impurities in super low abundance become clear. Like these 2 impurities, which I'll identify momentarily, at less than 0.2% of the main peak. These peaks have different retention times, but the same precursor of 659 and isotopic distribution. But I'll tell you, the increase of 16 AMU compared to the main peak indicate that these are isomers containing an oxygen atom. So to differentiate, we'll have to do MS-MS, and we'll get to that in a moment. But next is the impurity on the back end at less than 2%. And the sensitivity of this assay can be attributed to the xenotrap increasing the sensitivity of the system, ultimately ensuring that all of the sample you're introducing to the source is getting to the detector at the top of the TOF. This is the TOF-MS of that main MC3 peak that we just saw. The observed M/Z is for the singly charged MC3 with a single ppm, and we'll fragment this peak with a traditional technique called CID, as well as alternative fragmentation, the aforementioned EAD, or electron-activated dissociation. From CID, this inset here shows that it's not a very rich spectra. If you can see it, that there's any spectra at all. However, if we look at the same MS-MS data, but this time using EAD to fragment, then there is a wealth of information that we can gather with over 100 peaks that can be assigned to structures from in silico derived fragments. Now, more specifically, some of these peaks like 531, 545, 571, they can all be used to confirm localization of the double bonds at C6 or C31, C9, C28, and they'll both produce the 132 peak that indicates dimethylamino-butanoate head group, the one I showed you earlier. Only EAD produces the complementary fragment ions at 511, which represents the 2 identical alkyl chains. When talking about impurities, oxidation specifically, it's important to be able to localize the oxidation because of the study I mentioned earlier, which reported that N oxidations of ionizable lipids can lead to the loss of mRNA potency. So we have to be able to understand the oxidation to identify potential side effects. So thankfully, with electron-activated dissociation, we can do just that. So the first impurity we're going to look at is the alkyl chain oxidation, which would look a little something like this. This double bond is reduced to the alkyl group, or the double bond is reduced and the alkyl group becomes oxidized. Let's look at the MS-MS spectra. So at first glance, the 132 is present, and that's the one I said is indicative of the head group, suggesting the oxidation occurred on one of the alkyl chains. But where of those two locations that it could be located is what we have to find out. So with EAD, we're able to generate a few more diagnostic fragments that will help us localize the oxidation between C5, 32, C6, 31. So the epoxide proposed here is because of the lack of diagnostic fragment ions at 61 and 511, which would indicate oxidation of the head group. But with the oxidation occurring on the second alkyl chain, then you can see we are identifying these 558, 574, and 587 ions here. Let's take a look at what we'd see in that instance. So if the head group or the tertiary amine was oxidized instead of the alkyl chain, then we would observe this 61, 148, and 511.5. And you'll remember on the last slide at the bottom it said no 148. Another reason it's indicative that we were seeing the alkyl chain oxidation previously. But this is the impurity that we need to be wary of when developing our LNP formulations. If we go back to our MS-MS data, a unique ion at 61 indicated that the oxidation occurs on the head group. How are we certain that the oxidation doesn't occur in both places? You'll recall I said a mass shift of 16 AMU, so that's only one oxygen incorporation. But to confirm it even further, these diagnostic fragments here, 148, 511, show that the alkyl chains are untouched. So the oxidation could only be incorporated with the dimethylamino group itself. The last of the three impurities identified was the reduction of the double bond. However, there are 4 of them present on 2 different alkyl chains, so we need to localize exactly where the double bond has been reduced. So we'll look at the possible fragments for an unmodified MC3, which I've highlighted here in red, as well as compare them to what it would look like for a double bond modification after the reduction. This is an overlay of EAD MS-MS spectra of the main peak MC3 in red and the impurity peak that eluted at 16.9 minutes. That impurity peak showed a gain of about 2 AMU compared to the main peak of MC3. So fragmenting that impurity resulted in an increase of about 2 AMU compared to that of the main peak of MC3. Common fragment ions at 531, 545.4 are observed in both species and are indicative of the C9, C828 double bond. And unique ion pairs 558.5, 559.5, as well as 572.5, 573.5 indicate the location of one double bond reduction at the C6, C31. Taken together, this data demonstrates that the impurity was a result of a single double bond reduction by the gain of 2 protons, specifically at the C6, C31 location. And I'll give you a second to kind of take all of this in. For the sake of time, I've chosen not to go through each of the impurities identified in this workflow, but I wanted to provide you with a brief summary of the ones that we did cover and the ones that we didn't cover. But EAD was used to characterize the structures of the ionizable lipid MC3 and low-level related impurities. And it was achieved in a single injection LC-MS-MS experiment that used a 20-minute gradient. And compared to other fragmentation techniques, this product ion data is extremely comprehensive and provides an in-depth structural detail throughout the lipid species analyzed. MC3 oxidative impurities and determined specific sites of oxygen incorporation. And the data was also used to localize specific sites of double bonds and related saturation impurities. It has also been used, or EAD, I will say, has also been used to provide complete structural details of glycerol phospholipids and other ionizable lipids, not just MC3. The technique can be used to provide rapid, sensitive, and very detailed structural analysis of lipids within LNP formulations for liability studies and for raw materials testing. Overall, I would say the information provided by EAD can enhance product quality and safety of LNP formulated products, as well as guiding synthesis and development of novel ionizable lipids that have higher quality, stability, and maybe therapeutic efficacy to further unlock the real potential of RNA-based therapeutics and vaccines. I would like to take kind of one final minute to tell you about our SCIEX Now Learning Hub, where there is a lot of very educational content available just at your fingertips. Whether you've got whether you've just started in a new lab, or you've got new scientists starting in your lab and you're just looking for some great information that will get them started on capillary electrophoresis, mass spectrometry, or how to use either and more, please feel free to stop by our SCIEX Now Learning Hub. On behalf of Stephen and myself, I want to thank our 𝗕𝗫𝗵𝗿𝗫 speaker for joining us today and providing us with some of this great educational content. And we thank all of you for taking the time to join us today. It's been a lot of fun getting to learn mRNA and lipid nanoparticles along with all of you.
Thank you, Stephen, Paula, Nathan, and Dave for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. All right, so let's get started. Our first question here asks, what software do you need to process the 5' cap data on MS?
Thanks, Kaylee. So the simple answer is SCIEX-OS. This software has an intact quant add-in that is for automated deconvolution of a charge state series, and it was obviously originally developed for intact protein quant and flexible MAM, but it works just as well for oligos. You can set up the intact quant to focus on the specific capping intermediates to provide reconstructed peak areas of each, and then set up a simple custom calculation to calculate the % yield of the final cap product. So it is a bit of an automated software solution. And then additionally, you can also use Molecule Profiler to build custom 5' modifications of different capping intermediates. And this will help identify any related impurities to those peaks. But in short, SCIEX-OS can be used for both the acquisition and automated calculation of the % yield.
Great, thank you, Paula. Next question, will this slide deck be made available to participants?
I can take that one. So this is Stephen. I believe we'll have access to the on-demand version of this session, but feel free to contact myself or Paula if you'd like to get access to the slides. Our contact information should be available, and don't hesitate to follow up.
Great, thanks. And I'll add to that, Stephen, this presentation will be available for on-demand viewing after the session. And we'll continue with the questions here. This next one asks, can the plasmid DNA analysis workflow be applied to in-process samples?
And I'll take that one again. So it's really a great question because typically the biopharma industry tends to try to apply capillary electrophoresis for final plasmid drug product testing and not necessarily for in-process testing or monitoring. For in-process analysis, concentration is typically done by UV-Vis, and product quality analysis may be done by HPLC with something like ion exchange chromatography. There are some drawbacks to that method, though, specifically around assay sensitivity and its comparability to the gold standard assay, which is gel electrophoresis. So it could be manual gel electrophoresis for topoisoform analysis. That being said, we have had success with this workflow for in-process plasmid topoisoform analysis, and that's through the dilution of these crude samples, really to dilute out the interferences present in these crude samples. And we can do that because the assay by CGE, or capillary gel electrophoresis, really is sensitive enough to still detect these minor isoforms while being quantitative. So we do eliminate also the need for UV-Vis. So it can be a very nice in-process characterization solution.
Great. Well, we will go ahead and wrap up there. I want to thank you again, Stephen, Paula, Nathan, and Dave, for your time today and your important research. We would also like to thank LabRoots and our sponsor, SCIEX, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.